director of the Diabetes and Obesity Center. She received her Bachelor of Science with honors from the University of Leicester in the United Kingdom in 2006. And her interest in cardiovascular disease was initiated during her undergraduate time, as reflected in her undergraduate thesis, which was Responses of Cardiac Myocytes to Stimulated Metabolic Ischemia and Reperfusion and the Effects of Protective Interventions. She received her Ph.D. in Cardiovascular Sciences from the University of Leicester in 2011. And there her dissertation title was Diurnal Variation in Cardiac Excitation, Contraction Coupling, in Rat Ventricular Myocytes. That work resulted in 2 first author research papers in high-impact journals, Circulation Research and Journal of Hypertension. From 2011 to 12, she worked as a research assistant at the Open University and co-authored a paper that was a little bit different, I think, from her cardiovascular research, The Effect of Aging in the Internal Anal Sphincter. From 2012 to 19, Helen was a postdoctoral fellow in the Department of Pathology at the University of Alabama in Birmingham with Dr. John Chatham, where she pursued her interest in cardiovascular research there. She published 3 first author papers, a first author review, and 5 co-author papers, so she was highly productive. She currently has 3 additional papers with Dr. Chatham under review, so the saga continues. She has participated in a number of training opportunities in teaching and mentoring at UAB, and she's received a number of awards for her research. She's on the editorial board of 5 journals. I'm very impressed at that, because that's a lot. She's currently PI of a pilot project, Unraveling the Molecular Signature of Peripartum Cardiomyopathy, on a P30 grant to Steve Jones, and she's PI on a UofL School of Medicine Basic Science grant, Unraveling the Molecular Signature of Pregnancy-induced Cardiac Growth. So we're glad to hear about your work today. Thank you for speaking with us.
Thank you. Thank you for inviting me today to speak on my work that I've just started at the University of Louisville. And this will just kind of be like a taste of what I have got going on at the moment, and I'm sure if you invite me back in the future, there'll be plenty more to talk about. Okay, so I just have to disclose that, as Dr. Kling mentioned, I'm a pilot project PI on a COBRA grant. I'm also a co-investigator on this R01 here. I have a Jewish Heritage Fund for Excellence for faculty support, University of Louisville School of Medicine Basic Science grant, and IDEA Network funded proteomics voucher, of which I have 2. Okay, so I'll just speak to you a little bit about the research interests of the Collins Lab in the Diabetes and Obesity Center. So these interests encompass, first of all, investigating the sex differences in the regulation of the cardiovascular system and responses to pathophysiological stimuli, and also to investigate why there's increased risk of heart failure in vulnerable female populations, such as in postmenopausal women, Black and Latina women, those women with diabetes or hormone imbalances, or PCOS. Also, as the focus of today's presentation, to investigate the underlying signaling which contributes to the development of pregnancy-induced hypertrophy and peripartum cardiomyopathy. And it's important to study the mechanisms of cardiac growth during pregnancy and postpartum, because this can result in peripartum cardiomyopathy, and this is a leading cause of death in pregnant and postpartum women. And as you can see by this graphic here, that actually in the US, cardiovascular disease appears to be a predominant cause of maternal death in the US and also in the world. And so this reflects a unmet clinical need and a significant healthcare issue. So as we all know, During pregnancy, there are significant changes that occur within the cardiovascular system. These include an increase in cardiac output, specifically around 30 to 50%, which is mediated through increases in heart rate and stroke volume, as shown here on the left-hand side. There's also increased blood volume, which is also around 30 to 50%. There's reduced total peripheral resistance, decreases in systolic and diastolic blood pressure. There's an increase in oxygen consumption, and there's also, in addition to anatomical displacement of the heart, in which it's displaced upward and shifted to the, rotated to the left, there's also significant enlargement of the heart during pregnancy. So physiological cardiac growth occurs during pregnancy, and this growth differs from that of pathophysiological hypertrophy in that during pregnancy there's a normal or sometimes depressed cardiac function late in pregnancy, but for the most part this is normal cardiac function, whereas in pathological hypertrophy this is decreased. This hypertrophy is reversible during pregnancy. However, in comparing back to pathophysiological hypertrophy, that cardiac growth is irreversible. So usually also with pregnancy, there's no substantial induction of the fetal gene program. However, in pathophysiological hypertrophy, that's upregulated during that. And also during pregnancy, there's not any real documented fibrosis there or changes in capillary density and angiogenesis. It's fairly normal, whereas these are also changed substantially with pathophysiological hypertrophy. And so there are several signaling mechanisms which contribute to the physiological growth that occurs during pregnancy, which includes the PI3 kinase AKT signaling pathway, ERK pathway, and calcineurin. And although some of these, mainly ERK and calcineurin, have also been thought to contribute to pathophysiological hypertrophy and the signaling that gives rise to that, these two types of cardiac growth differ substantially. So there's many molecular factors which have been shown to contribute to the remodeling of the pregnant heart. Many of these are listed on the slide, and many of them include changes in metabolic proteins such as PDK4, PGC1 alpha, HIF1 alpha, the PPAR family. There's also, as I mentioned on the previous slide, documented changes in calcineurin. There's also some changes that are known to occur with some of the ionic channels in the heart during pregnancy. And also there's an, I apologize here for the VEGF, it's supposed to also have an upwards arrow, but there's been a documentation of both increases and decreases during pregnancy. We also, as we know, there's different circulating hormones and peptides which change during pregnancy, and many signaling markers that also change during pregnancy, in addition to many hormonal changes, such as those with respect to estrogen, progesterone, and prolactin, with some additional ones too, which will be shown in this slide here. So as I just mentioned, we get hormonal changes that occur during pregnancy. And with respect to progesterone, we see this increase in progesterone throughout pregnancy, which is reduced after delivery of the baby. We also see a somewhat similar trend with estradiol here. And we also see, although it's not shown in this figure here, we also see significant changes in prolactin. But also we see additional changes that occur during the final trimester and postpartum period with respect to the contributions of several circulating hormonal factors, which are undoubtedly will impact both systemic and cardiac function during pregnancy and postpartum. So we want to know what happens to the heart during pregnancy. So what happens if the heart cannot adapt to the demands of pregnancy? So for the most part, during a normal pregnancy, we observe a pregnancy-associated hypertrophy, which, as I previously mentioned, is reversible. So in this case, many of these, following the birth of the baby, the heart will, with time, regress back to its pre-pregnancy form. However, in some cases, this pregnancy-associated or induced hypertrophy can actually digress to a peripartum cardiomyopathy. And so there are several factors that are known to, or that have been postulated or have been shown to contribute to this change from pregnancy-induced hypertrophy to PPCM. But a lot of work is still trying to elucidate these factors. But with PPCM, some of the females that go on to develop this syndrome will also recover from this, but some of them will develop irreversible dysfunction and potentially require a transplantation or unfortunately pass away. However, so I'll give you a little bit more context with respect to peripartum cardiomyopathy. So it's an idiopathic systolic dysfunction which occurs usually during the last trimester of pregnancy or in the first 6 months postpartum. And as I alluded to in a previous slide, it's highly variable in terms of its severity. So we have a full range of the spectrum from recovery to end-stage heart failure. And in the US, this is affecting around 1 in 900 to 1 in 4,000 live births, but actually this rate is actually increasing year upon year, and that's thought to be due to females having children later in life and advanced maternal age. So as I mentioned in my first slide, cardiovascular disease and heart failure is the #1 killer in the postpartum period in the US. And as you'll see to the right-hand side, a lot of the incidence of PPCM, there's a high incidence in Africa and Asia, and this has been shown in the literature to be actually associated with increased salt consumption. Especially in Africa, the ladies there actually have a special diet that they're given after birth that's very high in salt. And then in North America, here, there's a fairly high frequency of PPCM, which is actually, the rate is actually in Kentucky is above the national average. So it's a very important process to study. So as I had alluded to previously, advanced maternal age is a contributing factor to the development of peripartum cardiomyopathy. You see here on the right-hand side that those females that are either older than 30 or 40 have an increased risk of developing peripartum cardiomyopathy. We also see with multiple pregnancies, the existence of preeclampsia is a risk factor. Pre-existing diabetes, also shown in this panel on the right-hand side, is actually more likely to, it has an increased incidence rates of PPCM in comparison to gestational diabetes. And this is also, diabetes is also a significant health concern in the southern US. Obesity, smoking, even cesarean section poses a risk of development of peripartum cardiomyopathy. And in terms of genetics, around 15% of women have a mutation in a cardiac structural or cytoskeletal protein, which will give a phenotype that's similar to dilated cardiomyopathy. But that doesn't account for the other 85% of people that go on to develop peripartum cardiomyopathy. So some of the proposed mechanisms of peripartum cardiomyopathy are still being elucidated right now, but some of those which have been documented include a cleavage of prolactin from this 23 kDa isoform to the 16 kDa isoform, which there's one significant treatment out there so far for peripartum cardiomyopathy, which is this bromocriptine, which blocks the release of prolactin, but it also, with that, the mothers are often told not to breastfeed. So then there's been other key players that have been implicated, such as disruptions in VEGF signaling, and also some STAT3 and PGC1 alpha signaling. And so, the molecular mechanisms which contribute to both pregnancy-induced hypertrophy and peripartum cardiomyopathy are not well understood. So understanding the mechanisms that actually contribute to what happens to the heart during a normal pregnancy will actually allow us in the future to effectively target peripartum cardiomyopathy. So in the lab, we've developed a pregnancy-induced hypertrophy model. For this, we've used the FEB strain of mice, 12-week-old females, and we have, for the purposes of the first part of the talk, we have 4 groups, which are control, non-pregnant diestrous females, mid-pregnant, which corresponds to day 8 of pregnancy, late pregnant, which corresponds to day 16 of pregnancy, and 1 week post-birth. And so these mice, for the purposes of this study, were subjected to normal chow through the study, and those which were in a pregnancy group at the beginning of the study, which were not pregnant at euthanasia, were excluded. And during these studies, we performed several assessments, the gravimetric assessments of heart weight, body weight, and heart weight to tibia length, some histology to examine myocyte cross-sectional area and capillary density, some echocardiography in collaboration with the echocardiography core in the Diabetes and Obesity Center, RNA-Seq analyses, and also some metabolomics and stable isotope metabolomics in collaboration with the mass spec core at the Diabetes and Obesity Center. So we see during, it's not surprising that during pregnancy we see an increase in body weight. We see which corresponds to which peaks in late pregnancy and corresponds to the point at where there's maximal fetal growth. We also see an increase in heart weight throughout pregnancy, which steadily increases until post-birth. And in some additional studies, which are not shown here, we show that even 2 weeks post-birth in this model, the heart weight is still increased. And we also, here we see a more definitive measure of cardiac size. We see here that the heart weight to tibia length ratio is increased throughout all stages of pregnancy and postpartum. And we also see an increase in myocyte cross-sectional area with respect to the late pregnancy time point, which is indicative of cardiomyocyte hypertrophy. So we next performed some analyses of cardiac function using serial echocardiography in collaboration with the EchoCore, and here we show, similar to our gravimetric data, we see an increase in LV mass at this late pregnant time point and post-birth. And for the purposes of this study, we didn't have the mid-pregnancy time point. We also see significant changes in the chamber dimensions of these hearts during pregnancy. We see that the end diastolic volume and the end systolic volume are both increased during late pregnancy and post-birth. And not surprising, we also see that an increase in both stroke volume and cardiac output during pregnancy. And as I alluded to in an earlier slide, we see a modest slight decrease in ejection fraction with respect to the post-birth time point. So what are the molecular mechanisms contributing to pregnancy-induced cardiac growth? During pregnancy, not only are there changes in systemic metabolism, but there are also changes in heart catabolism through changes through decreases in glucose catabolism and increases in fatty acid and ketone catabolism. So a recent study by the Irani lab examined this reduction in cardiac glucose catabolism during late pregnancy and found that there was a substantial increase in PDK4 expression, which is a pyruvate dehydrogenase kinase 4, which is an inhibitor of the pyruvate dehydrogenase enzyme here. And they also showed that during late pregnancy there was an increase in the phosphorylation of PDH. They also, as part of this study, showed that when you gave the PDK4 inhibitor, dichloroacetate, that you actually reversed the cardiac remodeling that was associated with pregnancy. And so the authors concluded from this study that a reduction in glucose oxidation was important for cardiac growth. And during this study, they also showed that the pregnancy hormone progesterone was responsible for that increase in PDK4 expression in the heart during pregnancy. We next went on to perform a RNA-Seq analysis on hearts obtained from our study animals. So for this particular study, we had non-pregnant, mid-pregnant, late pregnant, and 1 week post-birth mice, and we see with respect to the heat map on the right-hand side that there is a significant upregulation of proteins, gene transcripts during mid-pregnancy, and we also see a significant difference in the pattern in going from non-pregnant to mid-pregnant, mid-pregnant to late pregnant, and late pregnant to post-birth. And we see here that actually during late pregnancy, similar to that of the Irani study, we also see an increase in the transcript levels of PDK4 here. So we next went on to perform some supervised analyses of the RNA-Seq data. Here on the left-hand side, we have the PLSDA plot, or partial least squares discriminant analysis, which is a plot which shows the spread of the group separation between the transcripts that were identified. So we see here that between all groups analyzed that we have a good amount of group separation here. And so this here on the right-hand side is the VIP plot, which is the variable importance plot. And here, this is a plot that shows the specific transcripts giving rise to the group separation shown in this PLSDA plot. And you can see here that there's an we also see that PDK4 here is one of those genes contributing to the group separation. And we also, and then we also see several other genes of importance here in the VIP plot, such as HGM-GCS2 and MAP3K6 here, which are in important metabolic factors, which we will discuss moving on. So we next examined the top 50 differentially expressed genes and transcripts between the mid-pregnancy and non-pregnancy groups. And we see here that there's a significant upregulation in genes associated with cell motility, cell cycle, and cell migration. But we also see significant increases in those which are involved in angiogenesis and vascular development. So this highlights that maybe significant changes may be occurring with regard to the vascular compartment during pregnancy. And we have data to suggest that there's potentially differences that are occurring here because we also see significant changes in CD31 expression and markers of endothelial cell biology, which we will interrogate further in ongoing studies that we have in the lab. And so we next interrogated the top 50 differentially expressed transcripts with respect to the non-pregnant and late pregnant groups. And here we see there's several significant changes in metabolic genes during late pregnancy, which include this oxoglutarate dehydrogenase here, but also which include BDH1 or beta hydroxybutyrate dehydrogenase 1. And so why is BDH1 important? BDH1 is actually the enzyme which converts beta hydroxybutyrate to acetoacetate for ketone body oxidation. And so we actually see, although it wasn't shown on the previous slide, we see during mid-pregnancy, late pregnancy, and post-birth that BDH1 transcripts are increased. And we also see at the protein level, BDH1 is also increased in mid-pregnancy, late pregnancy, and post-birth. And of particular interest, additional analysis of BDH1, we showed that actually the transcript of BDH1 was highly correlated with VEGF-A. So these are particular studies which will move forward within the lab to interrogate this relationship. And in addition, as we next interrogated the top 50 differentially expressed transcripts between non-pregnancy and post-birth. And here, once again, we see BDH1 is increased, but we also see several genes that are increased that correspond to those of that correspond to the extracellular matrix and collagen changes here. And we see that a lot of these are also structural in nature. So we will be also interrogating the remodeling with respect to the post-birth time point, also in future studies in the lab to determine the significance of these changes. So in addition to the changes we observe in BDH1 expression during pregnancy, we also observed a significant upregulation of PDK4 expression in late pregnancy, and also a significant reduction in the phosphorylation of PFK2 at late pregnancy. And so these data here are suggestive of a reduction in glucose catabolism during a time point associated with the pregnancy-induced hypertrophy or the late pregnancy time point. And so this reduction in glucose catabolism at the level of both PFK and PDH could mediate coordination of ancillary biosynthetic pathway activity in order to spare glucose-derived carbons for both nucleotide and phospholipid synthesis. However, with the reduced reliance on glucose metabolism, the maternal heart must use an alternative substrate in order for energy metabolism to occur. So during pregnancy, there's a significant increase in the circulating levels of ketone bodies, both beta-hydroxybutyrate and acetoacetate, and these have been shown to be greatest during late pregnancy when this cardiac growth is substantial. And so ketone bodies are produced in the liver mitochondria and released into circulation and are metabolized in peripheral non-hepatic tissues. And in addition to their role as oxidative energy substrates, ketone bodies have also got non-oxidative, non-metabolic signaling roles, such as roles in acetylation and epigenetics and such, which would be interesting to examine moving forward. But the role of ketone body metabolism during pregnancy is completely unknown. So several recent studies have highlighted the importance of ketone body metabolism in cardiac growth and metabolic remodeling. In particular, studies involving both knockout and overexpression of cardiac beta-hydroxybutyrate have shown that not only can the heart use ketone bodies as a fuel source to maintain function, but that they can have a somewhat cardioprotective role here. And in addition, a recent study has shown that if you delete the mitochondrial pyruvate carrier in the heart and give these mice a ketogenic diet, that any pathology associated with that deletion is reversed. So these studies together highlight the importance of ketone body metabolism in the heart. So here's just a small schematic of ketone body metabolism. You have increases in fatty acid oxidation in the liver, and then you have a signaling cascade here, which involves the conversion of acetoacetyl-CoA by HMGCS2 to 3-hydroxy-3-methyl-glutarol-CoA, and that's then converted by HMGCl to acetoacetate. Acetoacetate can then enter the circulation itself and enter extrahepatic organs, or acetoacetate can be converted via BDH1 to beta-hydroxybutyrate, which can then enter circulation and also enter the extrahepatic organs. And then in terms of extrahepatic organs in terms of the heart, then beta-hydroxybutyrate is then reconverted by BDH1 back to acetoacetate, and then it's able to be used for energy production. But what's key to this is that BDH1 is not only important for liver or hepatic ketogenesis, but it's also important for ketone body oxidation in non-hepatic tissues. So of particular interest we see during in our pregnancy-induced hypertrophy studies that circulating levels of blood ketones are increased at late pregnancy time point. We also see, with respect to the late pregnancy time point, that there's a decrease in blood glucose levels, and that when you calculate the glucose to ketone index here, there's significant changes that are occurring with respect to the late pregnancy time point. So far we've established a model of pregnancy-induced hypertrophy. We've shown that mid-pregnancy is associated with changes in genes that regulate angiogenesis and vessel growth. We've shown that late pregnancy shows changes in several metabolic genes such as PDK4, BDH1, and oxoglutarate dehydrogenase. We've shown that ketone body utilization enzymes are increased in the heart during pregnancy and post-birth. That's with respect to BDH1. We've shown that glucose metabolism enzymes show changes that are consistent with a decrease in glucose catabolism, such as those that we have shown in PDK4 and PFK2. And we've shown that circulating ketone bodies are elevated in late pregnancy. So our hypothesis moving forward is that we believe that increases in ketone body metabolism are required to support the physiological growth that occurs in the heart during pregnancy. So moving forward with my studies, one of my aims will be to determine how liver ketogenesis contributes to cardiac growth and remodeling. So in order to do this, I'm in the process of generating, we'll mention my hypothesis first. So I believe that if we remove the contribution of liver-derived ketone bodies, that will show that these ketone bodies contribute to pregnancy-induced cardiac remodeling through an increase in biosynthetic pathways of glucose metabolism. So in order to do this, we're currently producing a liver-specific BDH1 knockout mouse in which hopefully the circulating levels of ketone bodies will be reduced, so we'll be able to assess whether these cardiac growth is impacted by this. Also, in another aim in the grant, we are hoping to elucidate the role of ketone body metabolism in the heart during pregnancy and how this may contribute to pregnancy-induced remodeling. So we believe here that increasing cardiac ketone body oxidation during pregnancy will augment biosynthetic pathway activity and growth signaling, which are both required for physiological cardiac growth. And so in order to do this, we're also generating the cardiomyocyte-specific BDH1 knockout mouse. And so we'll then assess the contribution of a reduction in ketone body oxidation to cardiac growth during pregnancy. So as I mentioned at the start, my lab is fairly new to Louisville, so we have several ongoing studies still, which maybe I can update you on fully in the future, but these include metabolomics studies, which will investigate the metabolites during cardiac metabolites that are changing during pregnancy and postpartum. In vivo stable isotope resolved metabolomics to examine carbon allocation and ancillary biosynthetic pathway activity during pregnancy. Plasma and cardiac proteomics studies, and obviously, as I described in the previous slide, moving forward with our liver and cardiac specific BDH1 knockout mouse models. So I have a small bit of preliminary data here from our in vivo stable isotope resolved metabolomic studies. So in this study, the groups may look a touch different as previous, but we have our non-pregnant diestrous group, mid-pregnant group, late pregnant group. And here we have our 1 week post-birth group But we also have here a 1 week post-birth with no lactation. So in order for this to occur, as soon as the pups were born, they were removed from the mum. So, and this will provide us some information with respect to those changes that are potentially driven by lactation or prolactin. So a brief description of the feeding protocol. So we have the day 1, and the mice are introduced to the liquid diet containing high glucose liquid diet here. And feeders are weighed, the food is weighed, and the mice are weighed. And then we come back 24 hours later, we weigh the mice, weigh the feeders, weigh the food to establish changes in weight and food consumption, and we replace this food again, once again with unlabeled liquid glucose diet. We come back 24 hours later, and then we weigh the food and the mice and the feeders again, but here we actually replace it with a C13 labeled glucose in this liquid diet, and then the mice are given this liquid diet for 18 hours, after which we once again weigh the feeders and the food and the mice to determine any changes in body weight or food consumption, and then we perform euthanasia and collect tissues. So we see, similar to the previous slides, that we have this increase in body weight during pregnancy, which declines following the birth of the pups. We do see a fairly consistent diet consumption with respect to the non-pregnant, mid-pregnant, late pregnant, and post-birth non-lactation groups. But this post-birth group with lactation consume at least double the amount of this diet in comparison to the other groups. We don't see any changes with respect to blood glucose, blood lactate, or blood ketone with this diet, and we don't see any changes with the number of pups here. With respect to other organs, we see a steady increase in liver weight during pregnancy, which is already, many of these parameters have already been documented in terms of the literature, and we see that with the removal of the litter that this liver weight dramatically reduces in size. We also see increase in kidney size during pregnancy, which is resolved with the removal of a litter. No change in the lung size, although we see some changes in the lung wet to dry ratio. These are fairly modest here. And we see, as also documented in the literature, some increases in spleen size during pregnancy. But what I guess the meat of this is during pregnancy, we show increased glucose-derived carbon allocation to both nucleotide and glycerophospholipid and amino acid to amino acids among other metabolites, which are not shown here, but and this is despite any changes in the metabolite pool. So these data here, which include, we see this UMP, glycerophosphoglycerol here, and L-methionine here, and these data suggest that an increased demand for biosynthetic pathway end products, which is met at least in part through ancillary biosynthetic pathways of glucose metabolism. And you see here, when you look on the graphs on the right-hand side, many of the metabolites, the highest fractional enrichment that we had seen was the best example here, I guess, is the methionine, but the highest fractional enrichment occurred in these post-birth groups, and this increase was reversed with the removal of the litter. So overall, these results suggest that several anabolic pathways that use glucose-derived precursors are increasing the heart during pregnancy and are potentially influenced by lactation. And this L-methionine is important because it feeds into this methionine cycle, which is part of one carbon metabolism. It's composed of the folate and methionine cycle. So in future studies, we'll investigate some of these downstream outputs to see if they're changed with respect to the heart during pregnancy. And just briefly, I'll quickly wrap up and just briefly touch on some of the plasma proteomics studies that we've done during pregnancy. And so here on the left-hand side, we have, this is not non-pregnant, mid-pregnant, late pregnant, and post-birth from left to right. And we see there's a significant increase in circulating plasma proteins in the late pregnancy time point. And we see over here on the right-hand side that there's with respect to this late pregnancy time point, this group seems to separate more so from the other groups. And so we see, with respect to this plasma proteomics, that we see there's certain documented changes that have been shown to occur in the literature, such as changes in alpha-fetoprotein, which is actually screened for during pregnancy, and that's increased in late pregnancy in our samples. We also see corticosteroid binding globulin, which is also increased in late pregnancy, and all these other three factors, which have also been documented to increase during pregnancy. So we're fairly confident that we're seeing some of the changes that have already been associated to occur during pregnancy in our studies. But of interest to me, we see the several isoforms of prolactin are increased in the late pregnancy time point during in the plasma proteomics studies during our late pregnancy time point, which is of particular interest. And we may use this kind of screening moving forward when we attempt to establish a PPCM model. Of particular interest as well, as I mentioned earlier on in the presentation, Cathepsin D has been associated with the cleavage of prolactin during PPCM. We don't really see the data for that in the plasma proteome is kind of variable. There's no real significant change there, but we do see other specific increases during either late pregnancy or also mid-pregnancy and post-birth in some of the cases of other isoforms of cathepsins. And this is actually important with respect to some of the changes, the remodeling changes that we're seeing, because some of the cathepsins are shown to be involved in some of the extracellular matrix remodeling. So in future studies, we'll investigate those further. But I think this is almost the last slide here. So we also performed another PLSDA plot, and we see a significant amount of group separation with respect to the plasma proteome. And with this VIP plot, which I mentioned, is a plot which shows the contributing factors to these group separations. We see here many of these cathepsins and prolactins, which give rise to these the group separation, in addition to several circulating growth factors. So these will be additional avenues of investigation at the laboratory moving forward. But so to conclude, pregnancy-induced cardiac growth involves coordination of several distinct signaling processes, and ketone bodies and other circulating factors may play a significant role in pregnancy-induced cardiac growth. However, many unanswered questions still remain. It's currently unknown whether there's a gene program that's activated and actually facilitates this cardiac growth during pregnancy. So hopefully some of the studies moving forward will interrogate this in more detail. Also, the extent to which other cell compartments play a role in pregnancy-induced remodeling is kind of lacking presently, my data would potentially suggest a coordination of several different cellular compartments, such as not only the cardiomyocyte, but maybe also endothelial cells and fibroblasts there. So to mediate not only the vascular changes, but the extracellular matrix remodeling changes. And also the full impact of the circulating hormones, the transient placenta, and maternal-fetal interactions on pregnancy-induced cardiac remodeling are still required in terms of the field. And the extent to which ketone bodies contribute to pregnancy-induced cardiac remodeling, and whether they even play a role in peripartum cardiomyopathy, is still an unanswered question, which I hope to answer in the lab. So I'd like to thank my faculty mentors in the Diabetes and Obesity Center, Brad Hill, Matt Nestoriak, Steve Jones, Aruni Bhatnagar, Shizuka Uchida, but he's moved on to a different institute, Sujit, the echocardiography core, which assisted with the echoes in these projects, Paul Lorkovich, who helped perform the stable isotope resolved metabolomics in the mass spec core, members of the Brad Hill lab, my former student Mallory, other members of the DOC, and I'd like to also thank my funding mechanisms, without which this won't be possible at the moment. And I'd like to thank you and take any questions, comments, and suggestions, and leave the code up for you.
So you mentioned a number of risk factors for the cardiac bad outcomes after birth. So I wondered if there's any protective mechanisms. And of course, exercise immediately comes to my mind. For women who are athletes, do they have a decreased incidence of adverse cardiac outcomes?
With respect to the PPCM literature, I don't feel as if there's not been an association there saying exercise is bad, but there's not also been, with respect to PPCM, I don't think there's been anything that says exercise has been good. There's nothing either way with respect to that. But there is a number of, with respect to the animal studies, there's a lean one study where she actually had exercised mice during pregnancy and found that basically the gene program was slightly different when you added exercise to that, and actually resulted in a further increase in some of the genes associated with the fetal gene program. So I would personally expect that exercise would be beneficial, but I personally haven't looked into that, but it's something that we might do moving forward.
Oh, I hear more. Are there any strain differences that are known in UWP? Now, of course, this is just, you're sort of looking at normal pregnancy, and I don't know if you've ever thought about, trying to understand pathological changes in the heart that occur during pregnancy in some animal models.
So the plan is actually to, so I had originally gone to interrogate our RNA-Seq data set with respect to one that Leslie Lean had done in C57s, and I actually see a slightly different gene profile than she does with my FEBs. So in the next couple of weeks, I've actually planned out a pregnancy study to interrogate the strain differences between the FEB mice and the C57 mice during pregnancy, because my also is required because my BDH1 mouse is on a C57 background.
Oh, right.
So I will need to move over, but the original studies were performed in FEB mice originally because the copulatory plug is much easier to identify. But we found moving forward that actually it's fairly okay in the C57. So that's in the pipeline, and hopefully I get to show some of that in the future, and hopefully the ketone body response would be similar.
How high are the ketone body levels during pregnancy?
So from what I'm aware, in human populations, I don't think that they routinely test for ketone bodies during pregnancy. It's my understanding that they usually test for ketone bodies if you're already diabetic and going into pregnancy, or develop gestational diabetes. But with respect to the literature, it's usually between somewhere between 0.5 millimolar to maybe around a maximum of like 5. But it's usually lower. I guess it depends on the duration of fasting as well, but typically the fasting response and the pregnancy response are fairly similar. It's only when you get up in the like ketoacidosis, which is like around 20 millimolar, that
yeah, but the ketoacidosis you really need to have significant insulin deficiency to get there. Because the reason I'm asking is, for gestational diabetes, you won't go into ketoacidosis. One of the things that we generally recommend, one of the ways by which gestational diabetics might try to control your blood sugar is by eating less. And especially when we ask them to check their ketone bodies in the morning, it will be very hot. And we tell them not to do that because, we want them to eat enough. Because, of course, they don't want to go on insulin, so they try not to eat. And because otherwise the sugar will be high, so they can put them on insulin. And the data on that has been in the past that having higher ketone bodies is not good for the fetus. Therefore, we don't want that to happen. So I don't know how this whole thing plays into what the maternal cardiac.
So the levels that we're seeing are like fairly normal. They're not too high. Like resting in the mouse, the resting ketone levels are around like 0, anywhere like a range between like 0.3 to 0.5 millimolar ketones, and the highest I had seen maybe during pregnancy was maybe about as much as 1.5 to. But there's also the balance with the glucose as well. So we see that.
So does it matter which ketone body? Is there a difference?
So at the moment, I've only assessed ketone bodies. So both acetoacetate, beta-hydroxybutyrate. So future studies, I'm going to assess both, but beta-hydroxybutyrate is the main ketone body. But it has been shown that acetoacetate will increase too. So maybe with pathology, the balance between the two is off and acetoacetate is much higher. But that's something I'll have to investigate moving forward.
Has anybody done studies with using an SGLT2 inhibitor to increase ketone bodies during pregnancy?
During pregnancy, I haven't seen that, no. I haven't seen that yet. That's something I actually had proposed to do in my COBRA, to either try a ketogenic diet or SGLT2 inhibitor, or maybe even a BDH1 overexpressor or something. Because actually, I don't know if I had mentioned it, but we're almost fully expecting this BDH1 knockout to have a PPCM phenotype. So that would be particularly interesting there. and there's a small amount of literature to suggest that maybe ketone levels are decreased with PPCM and preeclampsia at least. So I need to check if there's any. I had to stop sharing. There's one. Oh, it's the code.
Okay, so speaking of ketone bodies and going into the TCA cycle, didn't you say that there was a decrease in oxygen consumption?
Sometimes there is, and that was with respect to humans, yeah. Just overall. We have studies at the moment where we're actually interrogating mitochondrial function with the seahorse apparatus, and so therefore in those experiments we're providing the specific sex hormones, but also providing ketone bodies to see the effect on mitochondrial function. At present, it seems like it somewhat reduced its function, but I think the level which we have used with respect to ketone bodies was slightly on the higher side. So we have to go back and do some dose response there too, because I think I went in at 5. So that's probably a bit too much.
I have a question. It was a great overview, and we don't have that many researchers focus on pregnancy diabetes. So thanks for the nice talk. So my question was related to postpartum cardiomyopathy, which is really high mortality morbidity. So based on your data and everything, is there certain risk factors other than alluding to perhaps the ketone bodies and the overexpression that you were talking about? Is it more related to prediabetes, insulin resistance, obesity, hypertension, or any of those?
So I had mentioned that. So in terms of PPCM, 15% is known to be caused by women having a pre-existing mutation in a cardiac structural protein or somewhat. The other 85% is kind of tricky because it's hard to tell without maybe a specific biomarker as such as to know. Not all obese women will get peripartum cardiomyopathy. Not all diabetic women will. So it's tricky to figure out of which of those specific populations of women the precipitating reason. So there's several, like my list, I had a high salt diet.
I've seen it a little bit higher African Americans, at least when I was in Chicago.
Yeah, that's also a risk factor. Smoking, obesity, pre-existing diabetes, more so than gestational. Hypertension. There's so many factors, but if we can hone in on why specific women go on to develop peripartum cardiomyopathy, I think with perhaps maybe a biomarker as such, then that will be a significant advance with respect to that. And then because also unfortunately with these women, once they've had PPCM, they're also advised not to have pregnancies. Future pregnancies, they're also advised not to lactate as well, especially if they're given bromocriptine.
Yeah, some of the differences between lactating and non-lactating are very dramatic.
Mm-hmm. Yeah.
What the mechanisms.
I think it will yield some very exciting results, and probably.
For the most part, because we don't worry about prolactin much. You see a lot of people who have prolactin. They say, don't worry about it. Nothing will happen.
What was interesting to me is that in pregnancy, the weight gain can be dramatic, and if you look at a patient later in their lives coming in for obesity or weight management, they say the weight gain started during my first pregnancy. And, and then when you talk about ketone bodies and everything, that to me was like, kind of counterintuitive. But it's very interesting, that whole thing with the cardiac remodeling and the ketone bodies and all that.
Thank you.
So especially since, we have prolactin secreting tumors in men who have very high prolactin, and we tell them it's okay as long as, but if it has this effect, I don't know what happens in men.
Yeah, I hadn't. That would fall under one of the other branches of my investigations. So maybe there could be like a sex difference in response or something. So, yeah. Well, thank you for inviting me again. I can't. Thank you. Am I supposed to end the call?
Yeah.
Okay.